again. Hi, I'm back. Here's the next part. I wanted to get into this. This is where I wanted to show you guys that the cells and how they get the coronavirus is honestly a farce, because it's not. You just basically irradiated a fucking cell into like basically causing a cancer effect where it does force a mutation. Like, so you're not even looking at what you started with. It's just been bombarded with like chemical exposure and a bunch of other bullshit.
Virologists and ask what it is they are doing to claim contagious viruses exist and cause disease when zero real physical experiments in the world have ever been able to prove so. First, we need to ask, what is the cytopathic effect theory of virus particles? What is viral replication theory? Where do these unproven theories even come from? And what is a virus particle? This large area on the bottom left is the corner of a dying, rotting and decaying green monkey kidney tissue cell in a virologist's petri dish that has been starved of nutrients, poisoned, broken down, and decayed further with antibiotics like gentamicin.
They just chemically caused cancer. Like, they literally did everything and anything. Phenol. That's a poison.
Amphotericin, phenol red acid dye, and kidney-dissolving trypsin enzymes for many days. Then poisoned further with fixative neurotoxic preservatives like formalin, formaldehyde, glutaraldehyde, and then dehydrated in alcohol baths of ethanol and acid.
If you were to do this to any living thing ever, what would you get in the end? Like I said, an amorphous blob of like what used to be like considered like a living matter.
Acetone to drain water from the tissue sample, then put into a mold filled with epoxy resin, glue, and wax, then frozen to harden it, then sliced into micro-thin pieces, then stained with heavy metals made of lead and uranium acetate, and then bombarded with 150 to 300 degrees Celsius heat beams under an electron microscope to show us.
Just so you know, what's the sterilization temp? What temperature what temperature sanitizes?
For sanitizing with heat, water needs to be at least 171 degrees Fahrenheit.
I knew it was above $1.50. I only knew that because of working in the kitchen and shit. Because like when you clean stuff, you need to know how hot the water and shit have to be for doing certain stuff.
This picture of a still.
So whatever was in there is cooked to death. If it sterilizes bacteria, they killed it all. Everything's dead inside that. So whatever you're looking at right now, it might as well just went through a microwave.
Non-moving piece of cellular debris, which virologists label a virus particle. If you wanted to study and understand living human biology, would you study living active human beings, or would you dig up and dissect the corpse of a 3,000-year-old decayed mummy and just take a wild guess? To get an idea of just how small this chemically manufactured particle is, the smallest measurement on a ruler is 1 millimeter, and these particles are between 50 to 150 nanometers, or 0.
And I love bringing this up. And like I said before, one of the clips was, we have electron microscopes that can supposedly see the molecules, which are like frequency cymatics, looks cool, but like, we still can't see these fuckers?
0.0000.
I've seen videos of them zooming in on chips and shit, and the chips are smaller than these. They're
5 to 0.00015 of 1 millimeter. So what is this particle? It is a theoretical guess based on these kinds of electron microscope pictures that have been put through all of the ludicrous chemical procedures we have just described, and these pictures only. When we see pictures like this, these are just artist drawings and computer-generated pictures of what virologists...
This is what they do for space. Oh, I took the inputs of the thing, and then I used the coloring of what the electron... oh my god, spirometer, I think it's called. The thing that fucking reads it with the fucking radio frequency and shit. Microwaves. Electrospectrometer, I think it's called.
Think that black and white two-dimensional image might really look like. Nobody has ever seen one. It's imagination only. The theory is that it is this: a bunch of genetic RNA and DNA material containing instructions for making new copies of itself inside a host cell, with a viral envelope made from fatty lipid molecules.
Mind you, it's not supposed to be alive. So basically this thing floats around like living dander, but it's not living.
Taken from cells in the host with surface spike proteins that help the virus recognize and bind to cells in the host organism. You can see that too, right? It is not alive. It has no nucleus, no digestive system, no excretory system, no respiratory system, no circulatory system, no endocrine system, just genetic material wrapped in a ball of fat and protein. The cytopathic effects theory states that somehow this dead ball of genes, fat, and protein can find its way inside a person's cells and by some form of rainbow magic cause an enzymatic change in the structure of the host cell, even though
I like how they put all the triangles in front of our faces.
It has no enzymes to do so, which causes lysis, or the breakdown of the outer membrane of the cell, which blows the cell membrane open.
Well, you should be eating a nice amount of lycerin, like from your food. You wouldn't have these problems.
Killing the cell or rendering it unable to produce. The viral replication theory.
Once you get over the fear of being sick and you only have to worry about maybe exposure to certain things and chemicals and gases and stuff, life becomes a lot easier. It's not stress-free with a lot of stuff.
States that people sneeze, breathe, or cough these dead, lifeless balls of genes, fat, and protein through the air, and they somehow make their way into other people's cells. First, by attaching to cells via their spike proteins, and then using more unexplained rainbow magic to somehow penetrate the cell membrane, where they use more rainbow magic to somehow get the cell to make copies of them and explode them out of the cell.
So why don't they ever show a bunch of them at once?
Go do the same to other cells and other people.
Be that hard to find one.
This piece of candy is not alive. It has no nucleus, no digestive system, no excretory system, no respiratory system, no circulatory system, and no endocrine system. It's just genetic peanut and cocoa plant material wrapped in a ball of fat, sugar, and protein, just like a virus.
It is.
If someone told you this dead piece of candy could somehow make its own way into your garbage bag, use rainbow magic to multiply into a dozen more pieces of candy, and explode out of your garbage bag back onto your counter, you would probably call them insane.
Yeah.
And if they tried to...
Once you start looking at anything that has a theory, like the theory of gravity and stuff like that, when they put the word theory at the end of it, it's because it hasn't been proven. It's like quantum physics or something too. That's an easier way, because people like that a lot. Or black holes. It's all theories, quantum theory, because there's no actual proof, and they can't. It's just all theoretical. Trust me, bro. That's why it's trust me, bro shit. And they usually use the telltale sign of trust me, bro science is them leading you with jargon. Like if you start like on a basic thing and you ask them a simple question for, and they instead of giving you like an answer, they'll give you a whole bunch of big words that you have to figure out, because they'll hope you just go, oh yeah, okay, that makes sense.
Sell you a poisonous spray you could put on your countertop that they claim stop this candy from multiplying and covering your kitchen counter. You would most likely suspect a snake oil sales.
That's what they do, though. They make up the problem to sell you the fix. It's like I said, there's no new magic tricks. They just keep doing the same ones over and over again, hoping we're just retarded.
The first glaring problem with viral replication theory is that if it were true that these alleged particles go into the lungs, multiply, and explode out all over the...
There's a certain thing to that I can understand, because chlorine, but like... They're not. They're just straight up drinking shots of bleach.
Place, then it would be very easy to take a sick person's lung fluid, snot, sputum, prepare an electron microscope slide, and these particles should be found in there.
Yep.
After all, the lungs are a living, breathing tissue culture, or a living virologist petri dish, if you want to think of it that way. But the shocking truth is, these alleged virus particles have never been found in the fluids of human beings, sick or healthy. Virologists are taught in school that it can't be done. The particles are too small, and that's just not the way they do it.
There's not one published paper in the scientific literature finding this thing in any biological fluid of any sick human or animal.
You know what prevents getting sick? And he pointed out too. Is it him? I think he supports the idea. He's one of the people. I think I'm thinking of the other guy too, Berg or whatever it is. Smoking cigarettes or nicotine. Period. He even tells you like, don't really smoke it. Go for the patches more. But like, we have nicotine receptors, and they actually help us fucking a lot with our immune system, cancer, everything.
Anywhere. And everybody agrees. All the virus.
That's why they had a whole ad campaign. They lied about saying cancer, cigarettes cause cancer, and why they're trying to outlaw it everywhere. And they don't want you doing it because it's healthy for you. It's one of those big lies they tell us.
Virologists agree. They say that's not how you find a virus. We have 150 like institutions, the NIH and the CDC, etc. We asked them, can you show us a paper finding this particle in any fluid of any sick person? And they say we don't have such a paper.
Claim they don't have an isolate, but I mean,
they claim they cannot find the virus in any fluid. Now, even though they say there's a reason for that, because that's not how they do it. Now,
see, all those papers, like I was just trying to find that fungus one on one of the clips. They're putting it behind paywalls now, so you can't look it up. They don't want you to look at the PDFs, because my main thing during COVID, I had tons of them. Another weird thing that's happening, though, if anyone's been happening, Windows 11, the last update, they're complaining about Notepad. For some reason, my indexing on my file folders don't work anymore correctly. So, like, if the file had anything that says Epstein in it, it won't show up. I have to physically go into my folder and go search for them.
Here's the first question. Since the viral theory is I breathe in this virus, it uses my lungs as a culture, right? And so then it grows, and then it's teeming with viruses.
That means all viruses would be respiratory. If that was the case, too. They're not. By their logic, too.
That gets spewed out in the world. Why can't you find it in your lungs? It's a culture.
Oh, your point is if you do a direct electron microscopy of the fluid, you won't see this.
Never.
Okay, yeah, I see.
Never. Not once. We asked a guy who was 20 years, senior virologist at Wuhan Institute of Virology, 20 years head of Yale Virology Laboratory. Can you find SARS-CoV-2 in any fluid of any person you say has COVID? He said no. Why not? Because there's not enough virus to find. Okay, we asked him, what about if you put 10 people with supposed COVID and you mix their bronchial fluid together? Is there enough? No. 100 people? No. 1,000 people? No, not enough to see. 10,000 people, you mix their fluid together. Would you then have enough virus to see? And he said no, and then he wouldn't answer anymore.
But he claims what we see on the EM, on the electron microscopy slides, are in fact virus particles. The little bleb
what
things.
What he says is you have to culture them. There is no virus to see in your lungs,
right,
or in your snot.
Enough that they
Not
would admit.
anywhere.
Yeah.
So then what is a virology culture, and how is it they can see this alleged virus particle after a culture, but not in a sick person's snot? The technique was invented by a man called John Enders in the 1950s. Virologists take a person's snot, mucus, sputum, and put it in a petri dish that has dying, rotting kidney tissue from green monkeys. Then they add bovine calf serum,
See what I'm talking about?
like cow broth, sometimes milk.
There is so much nonsense they put in here. It is literally a magic spell.
And then they starve the fluid in that petri dish of nutrients so that the cells in the tissue start to break down and deteriorate. Then they help it along further by adding kidney-specific nephrotoxins, like the antibiotics gentamicin and amphotericin. And sometimes
Why would you give it antibiotics? If you said they're just trying to kill it.
kidney tissue dissolving enzyme called trypsin. Then they wait, and these chemicals break down the cells into millions of tiny particles, and then they do all of the other things we discussed already to prepare it for electron microscopy, and they say, aha, there it is, the virus particle. We can see it now.
Wait till they tell you how they make it.
There are obviously many problems with this procedure. First of all, there are multiple sources of genetic material inside a human snot or lung sputum, including lung tissue itself, debris.
The bacteria that lives in your lungs, they call it their there's a word for it now, microbiome, that's a bullshit thing. That's just them trying to get you okay to live with the fucking parasites inside you.
From dead human cells being excreted through the lung excretory system. Bacteria and microbes living in the lungs, food that went down the wrong pipe, inhaled plant pollen, RNA, and DNA from the air, inhaled bugs, and many more. Then the monkey kidney tissue itself has its own genetic material, as does the bovine calf serum, as does the milk, so there is no way to tell what genetic material came from what in the petri dish, or what you're even looking at under the electron microscope. It's just a giant soup mix of all sorts
I
of...
wouldn't even call it a soup mix. It's more like the scrap bin after, like, you ever seen those, like, the bellies for putting food scraps in to get, like, the methane from it, butane from it. How it congeals and becomes like a snot. That's what it sounds like.
Genetic material. So you can't even say where that particle came from. Is that particle from a virus?
Stupid.
Is it from the milk? Lung tissue? Monkey tissue? Bacteria? Plant pollen? Calf broth? Who knows? The second problem is electron microscopes take still pictures, not moving. Here is a man standing outside of his door with keys in his hand. Has he just come home and is going into his door? Or is he just leaving and just locked his door? You have no idea without the rest of the footage, to be sure. So did these particles go into that cell, multiply and explode out of it?
Why isn't there a bunch?
Or did the fact that the virologist just purposely put a bunch of chemicals in a petri dish that purposely destroy and break down the monkey kidney tissue, milk, bovine serum, human lung tissue, dead cell debris, bacteria, pollen, and other cells in that petri dish, and cause the cells to break down into millions of smaller garbage particles?
Honestly, what it looks like is a piece of pollen, and it's been, like, heated up a bunch and chemically burned. That's exactly what it looks like. It looks like
particles as the cell died, and that's what those particles really are: dead cell debris. Cytopathic effect is just a theory, because no person in history has ever, and I mean ever, observed that particle going into a cell, multiplying, killing that cell, and blowing out into millions of copies of the exact same particle, because that particle is too small to be seen without a still electron microscope photograph, period. Those particles have never been purified and isolated to even check that they are all copies of the exact same thing. Ever. No virologist disputes this. In fact, we are all told about autolysis, the breakdown of all or part of a cell or tissue by self-produced enzymes from the dying cell, not viruses. Here is a single-cell organism, just like any of your cells, experiencing regular death. And as we see, It too naturally breaks down into millions of nanoparticles of dead cell debris as it dies and deteriorates. That's what dying cells do. This leads to another glaring problem with virus theory. We are told about apoptosis, the natural programmed death of our cells that leads to the cell's nuclear fragmentation, meaning breaking apart into tiny pieces. Also, DNA fragmentation and mRNA decay, which would look something like this. Look familiar? We are told the average adult human loses between 50 and 70 billion cells out of its alleged 37 trillion each and every day. But are we sick every single day from the natural death of 70 billion cells? Of course not.
But because of fucking added toxins and shit, people start to feel fatigue and other stupid shit. Like it's aging. That's what he's describing at the same time. Is aging getting sick? Well, now, because people have been on the vaccine programs, yes. But, like...
Then where is the proof that a theoretical virus particle allegedly killing a mere fraction of those naturally dying cells causes any illness? There is none. This is not science. This is nonsense. Why on earth would virologists be using chemicals and enzymes like amphotericin, genomycin, and trypsin that cause cytopathic effect breakdown of monkey kidney cells to prove that a virus particle killed the cell and not the chemicals they use that do the exact same thing? It makes zero sense. And why are they using green monkey kidney tissue? Before we can examine and easily understand the errors in laboratory virology studies and procedures, we need to discuss and understand virus isolation and purification. Why is that so vital? Because if you are going to claim that that particle is a unique thing called a virus and not just normal garbage from a deteriorating cell, you have to prove it and then stick it in somebody and make them...
I didn't know what trypsin was. It spelled like trypsin. It's not toxic and stuff, typically in small quantities, but like so is most poison. But like if you inhale this stuff, it gives you asthma-like symptoms, and it doesn't irritate the skin. Wait, is generally safe, but can cause skin, eye, respiratory irritation, and potentially allergic reaction or asthma-like symptoms. So it's toxic. That's how you know they're lying when they can't just say, yes, it's toxic.
Stick with it, which has never, ever, ever been done in the history of the world. Ever. Suppose 3 people ate a chicken soup and all got sick with stomach cramps, vomiting, and diarrhea. And we had a theory it might be from the chicken in the soup. In order to be certain, first we'd have to isolate the chicken chunks only from the rest of the soup by putting it through a strainer to get most of the other soup ingredients and fluid out. Then we'd have to separate the chicken chunks only. That's isolation. Pretty simple. Then we'd have to wash those chicken chunks off with distilled water to make sure they aren't still coated with other liquids and ingredients from the soup. That's purification. These are not.
Hermetic.
Then we'd simply feed those people this isolated and purified chicken and see if they got sick with the exact same symptoms: stomach cramps, vomiting, and diarrhea. If they did, we have a good suspicion that it is the chicken, but we're still not 100% certain yet. We'd still have to run a control experiment where we feed them the rest of the soup without the chicken and see if they still get sick with stomach cramps, vomiting, and diarrhea. If so, it might be something from the broth or something else in the soup that the chicken chunks soaked up while they were in the soup. Fairly simple. But we're still not done yet. If it was the chicken chunks and not the control soup, we'd then have to run a genetic sequencing on that chicken to make sure it actually is chicken and not some substitute made of tofu, soybeans, corn syrup, and chemicals to make it smell and taste like chicken. So we're absolutely certain in the future that it was...
There's no refined fibers in those things, any of the plant-based shit, and like you need refined fiber. Not refined, natural fiber. You need it, and all the old food we used to eat used to contain it. None of it does now. It's all sterile.
Chicken that is the problem, and not some fake chemical substitute. Now let's say we want to prove that this particle labeled a virus actually is a unique thing called a virus and causes an illness. We'd simply take a sick person's snot or sputum, macerate it in a liquid solvent to break it apart a bit, pour that through a nanoparticle-sized mesh filter, just like a spaghetti strainer, to get rid of most of the larger debris bigger than the particle, so that we only have at least things of that particle size or smaller left in our sample. We put that filtered fluid in a density gradient centrifuge and spin it around until all of the materials in the test tube are in banded layers by weight and density. Just like your lighter socks and underwear stick to the washing machine wall, while the heavy jeans and shirts fall in the middle. Or just like these materials in this glass are separated by density. Then, we'd take a nanopipette and extract only that density band of material that only has these viral particles in them. And we have to genetically sequence them using either the Sanger method or the Maxim-Gilbert method to get the full genome, or see if that particle even has a full unique genome, to make certain we actually have some kind of unique virus particle and not just broken down partial fragments of normal everyday genetic material from the thousands of other things in the snot or sputum sample. In fact, this entire isolation and purification procedure would be even more important to do if you use the virologist's Petri dish culture instead of just taking the snot sputum directly from a sick person. Because now you'd have to separate, weed out, and eliminate all of the additional genetic material and whatever else they use to grow the culture. Then we simply inject or spray that isolated and purified virus particle material only into healthy people and see if those particles make them sick with the exact same symptoms as the sick person whose snot or sputum sample we used to get those particles from in the first place. That's isolation and purification. So simple a child could understand it. Now let's ask our virology experts.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and
I'm gonna rewind it. Okay, I'm back. Here's the other part to this. Start it from here.
That's isolation and purification. So simple.
Purification.
These are
it. Now let's ask our virology experts.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
Now, Dr. Gallo and Dr. Fauci talked a lot about isolation and purification. Can you tell me what the difference is between the two?
Isolation, what was it?
Isolation and purification.
When someone's gonna lie to you, that's the first thing they do is have you repeat the question again.
Of the virus?
What his last name is, like Hoffman or something?
Well, you isolate a virus by finding the virus which causes a disease. You purify a virus by making a lot of, just by purifying it so you get a pure virus.
Okay.
That doesn't even make sense.
I don't understand what the issue.
They interchanged the two, and I wasn't sure.
I see.
They have the fucking Caduceus in the background over there, big one.
If it was the same thing or if it was two totally different.
I don't know how they fucking managed
No, it depends on how they use it.
Okay. Can you explain the process of HIV isolation?
Well, I didn't Dr. Gallo do that? I mean, he actually isolated it. So,
Yeah.
I mean, why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. You know? I got other things to do.
I'm gonna pause this real quick. Hold on, let me make sure you guys can see this. What's on the belly of Baphomet? Anywhere he's depicted. Him and his tits.
Now, in their defense, it would appear that virologists are taught in their school and training that these virus particles are too small to isolate and purify and run a full Sanger or Maxim Gilbert genetic sequence to prove that it is a unique thing and not just partial genetic material from regular cellular debris. And that may have possibly been true before the 1970s, but that has not been the case for a long time now. And yet nobody has ever isolated any virus and sequenced an actual genome, let alone make a person or animal sick with the same symptoms by injecting or spraying those particles only with no other poisons, rotting flesh, or contaminants, ever. These so-called Petri dish virus particles that they see under electron microscopes are usually around 50 to 140 nanometers in diameter. Yet, when we look at a bacteriophage, they are anywhere from 28 to 200 nanometers in total height. And yet the capsid, where their genetic material is contained, is only 28 nanometers in diameter, much, much smaller than an entire virus particle. And yet scientists have isolated, purified them, and sequenced a full genome for them. Then we have exosomes, which are packets of protein, messenger RNA, and microRNA that are believed to assemble inside of a cell and then leave the cell to communicate with other cells in the area, among other functions. And exosomes are only 30 to 200 nanometers in diameter, nearly the exact same size as so-called 50 to 140 nanometer virus particles, and as you can see, nearly indistinguishable from alleged virus particles under electron microscopy. And yet, Dr. Andrew Kaufman sent me a paper showing that exosomes too have been isolated from blood plasma, purified through density gradient centrifuge, and even genetically sequenced. Which brings us to the nanotechnology industry and IBM, who has recently unveiled the world's first 2 nanometer chip technology for semiconductors, able to not only manufacture 2 nanometer
This is what I was thinking of when I think of like how small they can get, and they can zoom in on that. Is making chips, like I was saying, microchips, computer stuff. It's like
sheets, but also to attach them to nanochips. So let's put this in perspective. Are we really to believe that scientists can isolate, purify, and genetically sequence exosomes the same size as alleged virus particles, also isolate and genetically sequence the capsid of a bacteriophage that is literally the size of just 2 spike proteins on an alleged virus particle, and IBM can manufacture a 2 nanometer sheet 70 times smaller than an entire virus particle and attach it to a nanochip? But virologists can't find this giant particle in any person or animal's lungs or sputum, and even when they manufacture this particle in a petri dish, they can't then isolate, purify, and genetically sequence them? Really? It is painfully obvious that they are discouraged from doing so, because it will prove that these particles are not unique organisms called viruses, but just broken down fragments of dead cellular debris with identical partial genetic material from the broken down cells they come from. The pharmaceutical industry stands to lose tens of trillions of dollars in vaccine, cold and flu remedy profits, and over 20,000 prescription drugs.
Most of those drugs they give you for like being sick, like Benadryl and all that, they all just shut off hormones. Like aspirin shuts off hormones. It doesn't make your headache go away or actually reduce anything. No, it tells your brain, just don't release the hormones, bro. Let's see how that works out. There's a reason for the function to happen. It's your body taking you out. It's the putrefaction period before you become not sick.
To treat lifelong vaccine-caused allergies and illnesses. If the truth ever gets out to the public that viruses don't exist and the germ theory is a debunked fraud. What fraud? The purposeful creation of...
Yeah, there's 15 minutes left. I'll probably make a whole other video on that. I'll leave it in the background. Actually, while I'm at it, hold up. Make sure it's exactly where I left it off. I'm going to go edit all that together and put it together for the show thing. They're going to start doing this again. So get familiar with all this stuff. So when they start spewing out jargon towards us and telling us people are dying, we can just go, fuck you.